All right, we're going to go ahead and get started. So my name is Heather Wooden, and I am the IESC Meetings and Education Program Coordinator. So how this is going to work today, it's pretty much an open forum. So if anyone has any specific questions related to mold and fungal contamination, please, if you have a question, you can either unmute yourself and ask it verbally. If you don't want to interrupt, there is a raise hand option at the bottom of your screen. So what you would do is just click on that little hand and raise your hand, and then I will call on you to ask the question that you may have for our panelists. And then we also do have a chat box available. The chat box is located in the lower right hand corner of your screen. So I'm going to have everyone, we would like everyone to introduce themselves. So what I'm going to do is call on your name. Please let us know your name, company, and maybe how long you've been in the industry. Let's go ahead with that. So first on my list, I have, is it Girma? I'm sure that's probably not your real name, but you can go ahead and unmute. Let us know some information about you. All right, we'll go ahead and move on to Glenn. Glenn, go ahead. The unmute button is on the lower left-hand corner of the screen if you're there.
Did you call Glenn Rosecrans?
I did.
Okay, yeah. Well, I've been in contamination for 20 some odd years and working at Goddard. And some of our folks in our planetary protection group are concerned about the topic today. So I'm not sure if any of them are dialed in, but I always had this question anyways from being in the clean room and certainly with using alcohol to wipe off surfaces, that tends to do a pretty good number, but there's other places I haven't seen, and certainly the ductwork and such like that, that things can grow and fester. So that's it.
All right, thank you. Next, Jan.
I'm Jan Udy, and I am a fellow and president emeritus of the IEST. I also teach some of the courses for the IEST, and I am an independent clean room and contamination control consultant with almost 30 years of industry experience.
Okay, thank you. Next, we have Noelle. If I'm saying your name correctly, you can go ahead and
Yes, yeah, it is Noelle. Hi, I'm Noelle Long. I work with Cytiva. I've been in the industry mostly on the pharmaceutical side with aseptic manufacturing for over 30 years. And I am always interested in contamination control and keeping the patient safe.
All right, thank you. And let's try again. Is it Girma? You're done. You're at. You can go ahead and introduce yourself. It's like he's probably having technical issues. All right, so next we have up our panelists. So Morgan, why don't you go ahead and start it off with introductions?
I am Morgan Poland, and I've been in contamination control for over 35 years. And my latest iteration, I do a lot of consulting and help companies address contamination issues at Mycotoxin.
All right, then next I have Ziva. You can go ahead and introduce yourself.
Hi. Hello, my name is Ziva. I am a diehard mycologist, mold expert. My PhD is in mold, and I have worked and studied mold all my life. I'm also a med school dropout and also a clinical scientist. I am the founder and CEO of Microrite Inc., and we are a group of contamination control and other experts, and our goal is to prevent contamination. Of course, we do a lot of remediation and investigations, but our main goal is to prevent contamination proactively.
All right, thank you. And then last I have Jim.
Hey, thanks for having me here, Heather. And Ziva and Morgan are the very best. I just want to put kudos out there. It's always a pleasure to work with them anywhere, anytime. I'm also an expert in contamination control and especially cleaning and disinfection. I teach the course on that for PDA. I'm actually the PDA president for the Missouri Valley chapter, and it's a pleasure to be here today and have everyone here.
All right, thank you. Morgan, we did, and I don't know if you got it, but we did get a question through our website regarding this. So you want me to read the first question before we ask if anyone else has any questions?
Yeah, let's do that.
All right, so the question was, it's not really a specific question, but it's more about what the impact of filtration technology has on mold and fungal contamination. More of a topic regarding that.
So when it comes to clean rooms, for example, airborne mold, HEPA filters, the air conditioning system does a pretty good job at preventing it from getting into the clean room. And the whole aspect of HEPA filters are, per FDA, it to be at least 99.97% efficient at removing particles 0.3 micron and greater. And important to understand that that particle size when it comes to HEPA filters is the most penetrating particle size. So particles smaller than 0.3 micron actually don't get through either. So airborne mold, spores, and mold materials, Ziva can talk about that, can't get through the HEPA filters. Now, however, we can have issues. The oddball aspect of it we can have is if those HEPA filters get wet. And that's happened in facilities with high humidity, but mostly leaks. And then you can get mold growing in the HEPA filter medium. Depending on what the filter was filtering out, that can be food for the mold, and it can work its way through the fibers of at least fiberglass filters. I'm not aware of it happening in PTFE filters or Teflon-based filters, but yeah, that's my response. I don't know, Ziva, Jim, you have some, Jan, you want to expand on that?
I will take it from here if you don't mind.
Sure.
Yeah.
So, often we see, you have HEPA filtered incubators, and people don't look at, okay, they change their filters, whatever, every 6 months. What is inside the media is all the dust, dirt, your human-borne, the skin flakes and all. And if there is constant humidity, mold grows very fast. Now, if you have ever on your walk seen plants grow out of cement, correct? This is exactly what happens. The mycelium, that is the strand of the mold, can go through that point too, and everything from the back within the media is providing food, and that little mycelium then can grow into an aerial mycelium and produce spores. This is an issue that I have seen many, many times. People wonder, oh, you can have mold on the walls of incubators on the floor. No, it can grow in HEPA filters too, because mold, you have to understand, all living things adapt very well. And their purpose is to just sporulate and proliferate, and they will find ways. We have seen compromised filters, right, Morgan, cleanroom filters with mold growing. We have seen silicone around the filters having mold growing. So,
Yeah, and those typically the filters got wet or they were damp. They were, a normal HVAC system supplying clean air to a clean room. You shouldn't have those type of conditions normally, but what happens, the buildings get compromised or it's really humid. But yeah, we
One of
definitely
the
seen it.
problems, and I wanted to say one of the problems I see a lot, Ziva, in the cell and gene therapy arena is the gaskets on the incubators. Getting inside that gasket, you know, in the rib of the gasket, I see mold coming in there and being a problem, you know, like all the time. I know I work with Mark Glavkowski with a site, and it was cell and gene therapy, and they had big issues with that, you know, infecting not just the incubator, but when it gets out in the environment, then it's getting in the BSC hood. It's being, you know, spread around in the ISO 7 clean room, and it's a bigger problem.
Yeah. Yeah, I'm thinking, I
more-
remember having the issue in ductwork as well. So, like you said,
And-
dust stuff that's in there, it's food for the, and,
Yeah, and I want to add a mycology twist to this. I want you to know the same mold that grows on your bread mold, can actually eat your ductwork. And we have seen this in a pharmacy many years back. The ductwork was full of rhizopus and mucor, and that is normally you don't expect it to grow. So mold eats a lot of different substrates, right? They adapt, they change, they adapt. So anything that can grow where there is moisture or food, they will grow. I hope we answered the question.
Yeah, thank you. Does anyone that's on have any other situations or questions that they have for the panelists before we move on to some common questions that we get a lot? You can go ahead and just unmute.
Yeah, sure. This is Glenn. So obviously some of the duct work is very difficult to get into to actually clean, physically clean. So to kill it, you would probably have to use some sort of spray. Would it be an alcohol-based spray or something? I mean, if you do that, you're probably gonna
Create more issues.
fill your filters anyway, so you probably have
Yeah.
to remove your filters then after that process.
Keep your filters in good shape. That is what I,
Oh, okay.
Morgan.
Yeah, one of the
Yeah,
things
one
we
thing is-
see used a lot out there, and Ziva and Morgan have seen this, is VHP. So vaporized hydrogen peroxide. If they've tried everything else, so the liquid disinfectants, liquid sporicides, and they're still getting hits, a lot of times they'll move to VHP, and that can be spread around in the room. And if it gets in contact with the spores, it should kill it.
yeah, but for your ductwork, though, one thing is, you know, anything you do, you got to clean it, right? I mean, the issue is if you have dust in your ductwork and you're concerned about fungal contamination, VHP isn't going to get rid of that, right? You'll still have the dust, right? I mean, the point being is, you know, yeah, unfortunately, it's an issue, you know, in facilities, particularly as they age. You know, you get a buildup of contamination.
And that's a good point, Morgan. I think that you really should do the pre-cleaning with a surfactant-based product, whether it's a cleaner or disinfectant with surfactant, before you use a VHP application, if it's in ductwork or something like that.
Yeah.
I guess the trial there might be getting to it to clean it.
So difficult.
Yeah,
Okay.
duct cleaning robots. But yeah, I mean, whatever solution you do, it's going to require mechanical removal of the dirt. You know, some people attack the problem by going in and just, you know, people reaching their arms, shoulder length, their torsos in ductwork with a bottle of IPA and water. You know, and spray, and that.
Spray and pray.
Yeah, exactly. But you got to remove the stuff, right? So.
So Glenn, why do you ask this question? I am very curious.
Oh, no, it's just we got clean rooms there at Goddard that have been there 10, 20 years, and obviously some of the areas are difficult to reach, and you're going to have a difficulty in getting people to actually climb inside and do the physical cleaning. Some of the entry areas are slightly big enough, but you're still going to be limited by grates and bends and such in there that you're not going to be able to get to certain areas.
Sure. we don't see this very often, honestly. I've seen 2 cases where we had mucorrhizopus. Both of the cases we had mucor and rhizopus in the ductwork, but around the HEPA filters and anything that is open to interstitial space, Glenn, that is something you have to be very careful about, because interstitial space is full of everything, including critters, right? Um, and a lot of our mold contamination issues come because there's openings to, somehow there are openings to interstitial space, and then the mold comes down. And sometimes you will see mold chandeliers from ceiling tiles, right? Especially in the ISO 8 or the CNC areas. And you can literally see that there's mold growing, and it's all coming from the interstitial space.
Yeah, and what I see a lot too, Ziva, is pass-through decon. So especially in cell and gene therapy, items that are constantly passed through in there. A lot of times I see, like I did, audited a facility up in Montreal, and all they were using on it was IPA. You know, and the way they did it, they put all these plastic bags through an ISO 7 pass-through and into a rack, and the way they decontaminated like 100 bags, they just took a trigger spray bottle of IPA and sprayed it.
IPA
That's
is
really
a
common,
wishy-washy
actually.
disinfectant. Sorry, Morgan, go ahead.
Yeah, no, that's a very common way that companies do things. We do a lot of audits, we do smoke studies, we have to simulate all this stuff. And a lot of companies, they open up the pass-through, put something in, spray it down, and then close the pass-through, and then they wait. And that's a real difference in pass-throughs, between the US and Europe. GMP Annex 1 requires you to have an active pass-through, a pass-through with a HEPA filter, right? Where here in the US, in cell and gene therapies is notorious for this, where they have a static pass-through, and they'll be opening up from the ISO 7 into an uncontrolled space or CNC, passing material, and that air gets in the pass-through, and then they open that pass-through into the cleaner area, and that air moves. And we've had data that supports that might be one of the way companies transported fungal contamination through a pass-through. And like you said, isopropyl alcohol isn't going to kill mold spores necessarily. So, hey, Glenn, you have some real special concerns regarding microorganisms, particularly because you're supporting the Planetary Protection Agent initiative, right? So
I personally am not, but we have people that are. They worked on instrumentation that goes on one of the Mars gas chromatographs. And so they had a specialized clean room for doing all that work. Unfortunately, neither of the two folks that really know a lot about what they were doing in there are on. But in just general, we have a lot of other, that was a specialized clean room, and
sterile spacecraft.
yeah. And so our other areas where I've actually had just general concerns, they said, no, we're not going to put any assays in your clean room to then evaluate because, you're not going to get anything in there. And I said, well, I don't know. You just figure if we wipe it with IPA, it's all going to be gone, but we'll see. And granted, we're not, a lot of the items that we are wiping can get re-wiped periodically when we do inspections, but obviously we have never seen anything growing on any of our that I've talked with any of the 20 or 30 people that that have CC work at Goddard here, NASA Goddard, outside of DC, whether we have any issues or not with mold growing on anything, but I just want to see if there's any reason that we should implement some extra precautions along the way. And they had to do the same thing with Europa. They had to do a specialized cleaning with a lot of their propulsion stuff that's going to Venus. So they're worried about contaminating the next place it goes to versus how dirty it might be here.
I have a question then about that. My understanding is that any kind of environmental monitoring is not an absolute. It's a limited number of species that might grow if you pick them up. And then there's differences in media and incubation times. And I'm not necessarily certain that people might there's a lot of push to make it the shortest time at one temperature. And I don't necessarily know that folks realize they might be missing organisms. And how does mold, have you seen any of that with mold investigations?
So, I teach this course on mold contamination challenges through IEST. Uh, we've done it 2 years in a row, correct, Heather?
I think so.
Yeah. There is a big gap in understanding mold, and I've been teaching this for years. I've played with, as I told you, my PhD was on mold. I've played with thousands and thousands of mold isolates, and there is no end to learning. One of the things we don't understand about mold is not like bacteria. Mold is always evolving. So there are 4 types of mold. One is called the zygomycosis mold. That is your bread mold, to the family of the bread mold. It has 2 phases of reproduction. One, when it has ample food, it produces asexually. Those spores are weak. You can kill them to some extent with IPA and with phenolic or quaternary compounds. But when times are tough and food is not available, it produces sexual spores, which good luck killing. The moment times are good again, the sexual spore reproduces in an asexual manner. In the asexual reproduction, they create a lot more spores than in the sexual reproduction. But the asexually produced spores are much more easy to kill than the sexually reproduced spores. The second one is Ascomycosis fungi. They only have sexual reproduction and they have 2 layers of covering on them. I want you to understand that all the disinfectant label claim testing is done with weak spores, and the industry is waking up to the tough spores now, but it's hush hush. Nobody wants to talk to about. This is nobody's fault. Disinfectant manufacturers use AOAC testing. AOAC testing doesn't test with the difficult to clean spores. Coming back to Ascomycota, they have very tough spores. The third one is Basidiomycota, mushrooms. You shouldn't be seeing mushrooms and smurfs in the clean rooms, so we won't talk about them. And the fourth is Ditromycota, which reproduce asexually. Your aspergillus, your penicillium belong to that. Now those are asexual spores. Those are easy to kill. Remember that. They also have colored mycelia, the different type. There are 2 types. That is the sexually reproducing Ascomycota and the colored Ditromycota. They come from wood. So if you have mold, you cannot have a good investigation unless you know what type of mold it is, and that will tell you the source of contamination. One more tricky thing. The Ditromycota that I said last group always reproduces asexually. Over the past 50 years, we have realized that they switch. They switch also from not all of them, but we are discovering new ones every time. They switch from the asexual to the sexual spores. One thing remember, asexual easy to kill, sexual spores hard to kill. And if we keep that in mind, that will help us really have a good program for cleaning and disinfection and also for investigations. Am I making sense
Excellent
or did I
feedback.
confuse everybody? I wish I can share a picture here.
You probably can.
I don't know. I'll have to search amongst, but
Do you, is there any other questions before we move on? Anyone else have any other things they need to ask?
Well, Ziva, I just wanted to add something to what you said that I think is relevant here is back when I started in the industry here in 99, I can tell you that it was like a very primitive testing was done with fungal spores because I remember looking at a readout from a site in Texas and they said, well, Jim, it says black mold, yellow mold, pink mold, and red mold. I knew I'd get a chuckle. And today, if you showed that to Thomas Arista, he'd be like, well, I think we need a little bit more detail than that.
I want you to understand another thing. A good identification is worth it. If you don't identify it correctly, you will never be able to investigate it and never be able to remediate it. And I'm going to give you a case study here, which I really want you to listen carefully. I got a call from a company in the Far East say, what should we what should we use to disinfect people's feet? And I said, what? He said, we have fungus. I said, are you saying, why do you do that? They didn't talk to me. I happened to be on that site a year later, and I realized I was doing an audit, and I realized that they took a slide, a Scotch tape preparation, and they went on Google, compared pictures, and said it was a foot fungus. And the whole company and more companies started dipping people's feet in peracetic acid. Identification is very key to mold contamination investigations and remediation process.
One particular country has this practice at every single facility that we've been in that country. In fact, it's hundreds of thousands of dollars of equipment to automate and wash people's feet and hands before they go into the primary gowning area.
And your environmental mold is not coming from people's feet. They're pretty wrong ID will lead you to wrong. It'll be a wild goose chase.
Morgan, do you have any questions for our panelists?
Yeah, well, it's a comment. One thing about mold contamination, fungal contamination, it affects all industries that use clean rooms at some level, not just medical device. I sent a list of questions. I don't have them in front of me.
I can bring
Yes,
that up. I think I've got that. Let me take a look. I thought those were some good questions too, Morgan.
Yeah, I know. I was inspired at the moment. I can't recall them at the time.
Here, let me see. Okay. Do you want me just to go through them, or
Yeah,
so
please.
so one of them. Why is fungal contamination in clean rooms a different problem than other types of microbial contamination?
Who wants to take that on? Go ahead.
So, as I told you, fungi, the asexually reproducing fungi, are very easily disseminated by airflows. They are so light. Bacteria, yes, we shed bacteria or we have spore formers there on the soil. But if you have mold, it's very light and they produce much more spores than bacteria would. And they are very easily disseminated. So this one thing you have to understand. What is the point of ingress? How is it transported within the clean room, and whether or not it proliferates in the clean room? If there is ingress, it will come from the outside, and that's where we have the contamination control procedures, gowning, and removing wheelborne or foodborne contamination. But once if he gets into the clean room, your airflows are very important. If you are doing something aseptic work, and if you have an airflow where mold can go towards the aseptic, you will have mold contamination. We've seen it many, many times. The third is proliferation. This is when you see the same species again and again. That means it has found a home within the clean room, and now it is well settled and it is creating spores. So that is how you look at mold contamination.
All right, thank you. Jim, I have the questions up, so I'll go ahead and ask them now.
Okay, sure.
Oh, Morgan, did you have something to add? I thought I
No,
no.
but yeah.
Okay. So some people, another question that we see is we fogged our clean room, but the mold keeps coming back. Why?
Because you don't know what the ingress point is. And to ingress point, again, understanding ingress transport is very important. We do so many mold contamination investigations, and we have a methodology to look at ingress points, and that starts with understanding what mold species you have. Molds, and I'll give you a very good example. This is the latest example Morgan and myself have. They had all kind of mold issues. They've had it for a long time. It went into the fermenter, whatnot. And they sent me the list of IDs. And I said, guys, you have insects. And they never believed me. I said, you have entomogenous fungi. Entomogenous is insect-related fungi. It couldn't be. We walked through the facility and we showed them where the ingress point of insects were. So to understand the ingress point, first the mold type, then the ingress point, and then you tackle it and you can resolve the issue.
And
The
I want
pest
to
control
add,
is part of that.
I want to throw something out there too on this discussion that Ziva just talked about before that, is it's really critical to ID the mold species. Because when I was up in Montreal for a audit a few years, before this whole pandemic happened, I can tell you that there, when I went on site, they said they think they have a mold contamination. They're picking up a black mold in the mulch pile in the parking lot. They're picking up a black mold in the sterility testing suite and a black mold in the ISO 5 clean room. And I said, well, with this black mold, do we know exactly what type of mold this is in all three areas? And they said, well, we never thought to check that. How important is that, Ziva?
I am big on IDs because I've done IDs a lot. And without having a solid ID, you cannot investigate. Gone are the days of black, green, and blue mold. You have to get, things are changing so rapidly in the mold world, and you guys are not entrenched in it. I am. The whole Amsterdam Convention has changed the taxonomy and the nomenclature and the classification of mold. And what people don't understand, and this will be good information for Noelle because she works in aseptic. Um, we get IDs now in the sexual phase. The sexual phase is hard to clean, but the asexual phase of that mold is clinically relevant. So if you don't know, if you get an ID with the sexual phase, and if you don't know that this mold has an asexual phase, and you say, oh, no, there was no infection with this mold, you might be releasing adulterated product. So ID and understanding the phases of growth of mold and the methods of reproduction is very important. I've been teaching this hands-on course for many years, right? And I won't stop teaching till people understand that unless you understand the mold, and as Jim said, unless you know the ID, you should not move forward with the investigation or remediation efforts. I hope I'm not scaring you guys.
We don't scare
Thanks,
so
you
easy.
guys.
Morgan scares more than me.
So let's say the FDA comes into your clean room and they make a comment about rust. Why?
Morgan.
Yeah, it means you're probably using bleach, right? Facilities that we get involved with that have issues with fungal contamination. One of the common symptoms we see when we go to the facilities is the floors degraded, and you see rust on areas. And that's a sign. Hey, you got I go to a facility and I look around. If I start to see a lot of rust on wheels and low level things and things around in the clean room, I'll ask, do you guys have a problem with mold contamination? And you push them enough, or you're there enough, and eventually comes out, yeah. And have you guys been using bleach? Right? It's often commonly used, and that's why you see that.
Mm-hmm.
Yeah, and I can tell you guys that I hear a lot from facilities that come to me to ask about compatibility, and they'll come in and they send me so for example, flooring surfaces, and many times with stairs, oh, it's aluminum. Not
Yeah.
Not a very good surface for bleach.
Mm-hmm.
Yeah.
True. And Jim, you brought up such a good point. People never see people never bring up or understand the compatibility issues when they develop a disinfectant program, because certain things are not compatible. I can give you a lovely example of a mold and disinfectant. So the company had Ketomium. Ketomium is the one that produces sexually, has 2 layers of protection, right? Well, they had on a filling on a in the filling on the filling line, right? So the director said, oh, sporclans or whatever parasitic acid, if I double the concentration, I will be able to kill the mold, because at normal concentration I cannot. What happened? That compatibility was at the concentration that the manufacturer had tested it for. The moment he doubled the concentration, all the walls were eaten. So
That's exactly right. I believe it.
it happens all the time. And of course, they were then scared of the parasitic acid chemistry to use. And I had to tell you, you are PTSD to parasitic acid. You might need some therapy.
Yeah, and sometimes we found a good example, Ziva, is we found in some cases with certain organisms outside of mold, a good example would probably be Bacillus cereus. You may need a two-step process to kill it. You may need a cleaner with bleach in it, followed by a parasitic acid product, because some organisms can be extremely hard to kill.
Yeah, and you are lucky if you get a 2-log reduction with B Series.
That's the
Yeah.
reason it's serious.
Yeah, and you are lucky if you can. You will not be able to kill Ascomycosis fungi. That is why it is important to understand, even if you have a Deuteromycosis fungi, which is easy to kill, if it has an Ascomycosis stage, you better be prepared for a good long remediation process, because that double layer of protection, it's very hard to kill.
And I
So
I
can
had a
attest to that.
question.
Go ahead.
Is fungal contamination issue in non-sterile, internally sterilized products and medical devices?
Absolutely. So for non-sterile product, it is very, so I don't know how many people have seen the latest non-sterile guidance by FDA. This is an issue that we don't understand. We go by the USP um, USP 1111, and we say in a nasal product we will not have staph or pseudomonas. In a inhalation product we won't have staph or pseudomonas. In a dermal product we won't have staph or pseudomonas, or maybe candida. Actually, the most emerging infections are from mold in sterile and non-sterile products. And a good example is, uh, I don't know how many of you have read about the mucormycosis that happened in India to the people who actually recovered from COVID. It was never such a big issue, but mold looks for weak immune system. Mold does not have endotoxins or exotoxins by which it infects. Mold literally physically attaches to the soft tissue and spreads. So non-sterile and I teach a course on objectionable microorganisms, pathogen specific assessments, and I've shown how many mold are more infectious than the limited amount of testing that we do. And good example is the New England pharmacy. 3 mold were discovered. 2 of the mold had both sexual and asexual. No wonder they could not kill it, because they were switching. Oh, the mold in non-sterile should be considered seriously, and FDA has touched on this point in their new guidance.
What
And
about
I can,
terminally
I
sterile
can, I was
products?
going to say, I can add to that. Ziva, I actually was in, before the pandemic, I was down in Rio de Janeiro, where they did the same practice that they were doing at New England Compounding Pharmacy. So this pharmaceutical facility was shutting down the air handling system at night. And it's very hot and humid there. And then the lady looks at me and she says, "How do you think we've got such a mold problem?" And I'm like, well, when you look in the rooms, you look up at the ceiling and you see these black spots everywhere. And I said, I don't think that that's the normal color of the ceiling.
Oh yeah, we've seen this.
My
Too.
question to extend that, what about terminally sterilized products? Can mold be an issue?
So I will start with an example. One of the medical device company, which had a lot of lawsuits, they called me. They said, we have chetomium in our sterility test. Okay, it means sterility test failure with chetomium. So it is the lab, and I knew the lab. I knew the owner. I said, I've audited this lab twice this year. This is not the lab. Where do you manufacture? So they said, oh, we have a CMO. So I said, let's go to the CMO. The CMO, the clean room, their clean rooms were perfect. Such good clean rooms. And I said, so now you make this medical device, you wash it, right? You dry it before sterilization. Where do you do the washing and the drying? Where do you think they took me? To the warehouse, a little room in the warehouse with an air conditioner. And, the warehouse is full of pallets. And this chetomium that I'm talking about, Ascomycosis fungi, comes on pallets, on paper, wood, all that. All I had to do is take a swab from the air conditioner, and there it was. It was blowing right on where they were washing the product. And I'm doing a course at STEC on how to kill bacteria and fungi. This is something that we are discovering, and you have to understand that our BIs are much easier to kill than some of the tough mold. So you, in medical devices too, you have to be careful as you are in in any other medical products. Morgan, did I answer the question?
Yes, I think so.
Well, I think a follow-up question to that would be that if my product is terminally sterilized, like bacteria can leave endotoxin. I've read that molds can leave like mycotoxin. Is that an issue?
So I love that topic of mycotoxin, Noel. So most of the mycotoxin studies were done in the agricultural field over a lifespan of 70 years. There is mycotoxin testing. I don't think it's a very big issue. Uh, like endotoxin too. You have 1 or 2 gram negatives, it's undetectable, right? You have 1 or 2 mole, it's undetectable. But in the non-sterile, the challenge becomes if you have mold in your non-sterile product, and if it facility, and if it gets into your product, your antimicrobial efficacy testing or preservative efficacy testing is weak, like I talked about the disinfectant label claim, right? So what do we test it with? Aspergillus. And now you have a tougher mold to grow that can easily grow circumventing the antimicrobial. We have seen this many, many times. And at that level of mold, mycotoxins could be a product. But you do get mycotoxin testing kits from Associates of Cape Cod, and who are the other ones, Jim, who make endotoxin and mycotoxin?
Let's
Endosafe.
see. Does Charles River? No?
Yeah, Charles River does
Charles
that.
River, yeah.
So, and I'll give you an example of this. There was a IBD company, this was many years back, said, hey, you know, we have these reagent cuvettes. There are fuzzballs in that. And when we went there, I mean, that facility was in a very bad shape. They had cladosporium, urosium, all these kinds of mold. They hadn't cleaned the clean rooms, I mean, cold rooms for 12 years. They had 10 or 12 cold rooms, literally fungal chandeliers hanging from the ducting because the ducting is wet. And their antimicrobial in the reagent was circumvented, that efficacy was circumvented by this mold because the testing was done with an easier to prevent mold, that was aspergillus. So you have to look not only as you know, will it get sterilized? Will it proliferate in your product? But can your antimicrobial stop it from growing in the product? Noel, did I answer your question?
Yes, and more. Thank you.
You're welcome. You see, I'm crazy about mold, right?
I love that.
So what actually kills mold? Does alcohol kill mold?
Prayers. Alcohol does not kill mold. Alcohol is a wishy-washy disinfectant. According to EPA and FDA, it is an agent that can reduce the number of organisms per as acceptable by public health ordinance. This is the literal definition of alcohol, which is a sanitizer. Who knows what public health ordinance is? Some phenolic and Jim, you can chime in, and quaternary ammonium compound will kill the easy to kill mold because they're tested with easy to kill mold like Penicillium, Aspergillus, Bovaria, Pseudomyces, and all that, Acremonium, so on. The Ascomycosis fungi are very hard to kill. If they switch from sexual to asexual, in the asexual stage, you can kill them. In the sexual stage, they're hard to kill. Jim, did I say it right?
Yeah, so I would say, as you were saying, it depends on the mold species. But what we see typically is some of these next generation phenolic disinfectants, next generation quats will have some efficacy against some of the molds. Potentially aspergillus might be one, which is hard to kill. As Ziva said, when you talk about molds like Cotomium, then we start to talk about sporicides like bleach at high concentrations or acidified bleach or hydrogen peroxide peracetic acid chemistries for an extended contact time. Those come to mind, and you're exactly right about alcohol. The biggest problem with alcohol is if I spray a hard surface with it, it will basically evaporate in 30 seconds or less. So it evaporates very quickly. So in order to get that wet contact time, it's very hard with IPA or ethanol. These,
And it doesn't kill.
formulated disinfectants, it's not.
And
Sorry,
this
I
is
didn't mean to interrupt
it's
you.
an industry. We are learning so much about mold now. Uh, and this is something that I knew all along, that people are going to, you know, put all the mold in the same basket. You cannot. You cannot. Morgan?
I definitely agree. With there's those cleanliness to get rid of mold in facilities, cleanliness would have to be really important, right? I mean, in terms of keeping things clean, getting debris off the floor. One of the things I see a lot in facilities are I look a lot at floors. do a lot of smoke studies, one of the things we do is we see how the smoke moves through the facility, and you'll see HEPA filtered air come and strike the floor, and then you get this bounce back. And when you start to see around the perimeter of the building or the room you're working in, you sometimes will see hard to clean areas. A lot of companies still use door sweeps on their doors, and they're never cleaned. And a lot of these have individual strands. Some are still horsehair strands, if you can imagine, or plastic fibers, and they're broken apart and they retain a lot of stuff. And that's a place where mold can prosper. And what happens is, as people, workers are walking, they're wearing sterile garments or shoes or booties, whatever, and they track that in further into the room. And what happens is, if floors aren't kept immaculate, you can get movement from the floor up along an operator's body in some case. We've seen smoke studies where the air, the suction of the RABs, the enclosure is so strong, air is being pulled from the floor up over an operator. And what happens, they're standing in their sterile gowns, and you have this upwards movement of air from the floor over the operator's hands. And during some interventions or equipment setup, they'll be reaching into the grade A or the ISO 5 manufacturing critical zone. And this is how this stuff can get tracked in. And if you have some and this is the issue with isolators, people think isolators are magic boxes where you load a bunch of stuff in the beginning of the campaign, extra tools, utensils, wipers, plates, EM plates, or those things in small assemblies. They close the isolator up, they hit it for the decontamination cycle, and it's really good. But if in that setup situation, if you're tracking in some of these harder to kill mold species, that can get into the product. And we've seen that companies have had those issues. And when you find out, what happens is companies back down. Used to be a grade B environment, basically meant ISO class 6, right? And companies got a little bit cheaper, and they're now the ISO class 6 design rules, right? The operator likes ISO class 7. But companies have backed off from that, so now they're under ISO class 7 design rules, and then with grade C, ISO class 8 design rules some cases, and you don't have a really good background environment. And again, all boxes, isolators, even the greatest isolators today have to be opened up during setup, during configuration. That's how this stuff can still be is an issue.
Morgan, I want to give the example of this terminally sterilized. And, people think terminally sterilized, everything is killed. This terminally sterilized company, and they had like 3 or 4 warning letters. And the same thing, you had a leak in the ceiling. But when we visited and did the investigation, there were 2 problems. Once they had the leak in the ceiling leaks means all your walls and the ceilings are prone to mold contamination, right? So now the mold contamination is in the grade B, grade C area, right? And as Morgan said, the RABs that they had, the inlet suction was so strong and it was next to the grade C HEPA filter. So all the HEPA filtered air which was coming to the grade C, supposed to clean the grade C, was getting sucked into the inlet of the RABs. That means the grade C area was not getting cleaned. And now the mold from the ceiling, compromised ceiling, is falling on the people. People are laden with mold. Now they're going into the RABs grade A, and they're introducing mold into the product. And they thought that terminal sterilization will kill everything. And no, they had mold in their terminally sterilized product. So there are multiple aspects. What did you say?
They're optical products. They're optical products.
Yep.
Does anyone have any other last questions before we end this webinar? Anything from Glenn, Noel, Jan?
So just a question which I've seen in the aerospace industry, particularly talking about sterile spacecraft, an argument between equipment engineers wanting to use 99% isopropyl alcohol to wipe down the spacecraft, and the microbiologist attached to the group, saying, no, it's got to be 70% isopropyl alcohol. Jim, Ziva, do you have a comment on that? I mean, my understanding was you need 70%. It shouldn't be any stronger. But I'm not a microbiologist.
Yeah, let me give you my feedback, then I'll turn it to Ziva. You really need the 70% because you have to have that 30% water, because that's what's needed to get through the cell membrane and the cell wall into the bacteria or the fungal cell to essentially lead to the rapid cell death. So you need that amount of water to be uptaked through the receptor sites to get into the organism. If you go with, say, 99% alcohol or 13%, you don't have enough of that water to get the migration through the cell wall, cell membrane to kill the cell. And where that's evident, if you look in the Seymour S. Block book on disinfection, you'll see that they have a really nice table in the alcohol section that shows the titration of different amounts of alcohol, ethanol, and isopropanol. And you can see that the sweet spot for efficacy is 70%. Go ahead, Ziva, sorry.
Oh, no, you covered it. All I want to say, you go beyond 70%, the cell feels like after a couple of margaritas, but it's not dead.
That's what we need now is a couple margaritas.
Yeah.
Ah,
Well, Glenn, do you have anything to say? Question?
No, that was very good for the question there regarding the use of 70%. We have recommended that for gross cleaning, but it's also, now I know that it's really good for also for its lingering time and the helping out with disinfecting the commonly handled GSC that is used in clean rooms, ground support equipment, harnesses, the racks. People are touching them with bare hands a lot, and so certainly there's a lot of transfer contamination there.
Noelle, did you have something real quick?
I don't know if it can be answered quickly, but just about the recommended incubation time and temperature for mold recovery.
yeah, I
And
can
if
tell you.
Go ahead.
So mold, see, it's mesophilic, right? It grows great. And so this is a question. Okay, I'll try to answer it. So I have never understood why we suggest, and I've had my own challenge with USP. Why are we incubating bacteria at 30 to 35 and mold at 20 to 25? Mold is, mold grows more in tropical regions and subtropical than in temperate regions, right? That being said, I am a big proponent of doing a study, and you can do the study to show that the same mold species that you recover at 30 to 35 can be recovered at 30 to 35. As far as the days are concerned, do 5 days, 7 days, it will take over the plate, especially the Deuteromycosis fungi, and you will not be able to count colonies. Noel, you can write to me. I can explain to you better if you need to. It has to go through Heather. But this is a challenge in the industry, and it's also, I mean, I have a very different opinion from years and years of experience.
Thank you.
Yeah, thanks everyone. Thanks Ziva, Jim. I know Morgan had to get off the line for everything today. And thanks everyone for participating and all your questions that you have. Yeah, and if you guys have any other additional questions or something, you can go ahead and just email me. And I can forward it on to Ziva. She's able to answer any of those questions. But yeah, thanks everyone for participating, and everyone have a good rest of their day. Thank you.
Thank you, everybody.
Thank you. Take care.
Bye.